very pleased to have Dr. Jeffrey Millman here with us. He's an associate professor of medicine and biomedical engineering at WashU School of Medicine. And his talk, oops, sorry, just letting more people in. Today is Unraveling Cellular Complexity in Stem Cell-Derived Islet Identity through Single-Cell Sequencing. Just a little bit about him. We last had him sort of on Sugar Science. He was one of our very first speakers, actually, episode 11. He did a podcast for us, August 27th, 2020. Sort of right in the middle of COVID, and he was just so graceful to take some time and talk us through his work, which at that point was very interesting. And now he's come out with this fabulous paper, Comparative Integrative Single Cell Analysis Reveals New Insights into the Transcriptional Immaturity of Stem Cell-Derived Beta Cells, published January 24th, 2024. First author, Mason Schmidt, who's also on the call today. And just a little, sorry, a lot of people coming in. Just a little bit about Dr. Millman. He's an associate professor of medicine and biomedical engineering at WashU School of Medicine. He received his PhD from MIT and completed his postdoctoral training at Harvard University. His current research is focused on synergizing both biomedical engineering and cell biology approaches to use stem cells for the study and treatment of diabetes. And he's developing new approaches for the differentiation of insulin-producing islets from stem cells and improving the utility for cell replacement therapy in vitro disease models. His innovations have been licensed to biotechnology companies in the diabetes space and resulted in ongoing clinical trials. And he previously served as the vice president of research at Sana Bio, where he oversaw the preclinical development of hypoimmune stem cell-derived islets for the treatment of type 1 diabetes. His laboratory is supported by the NIH, NIDDK, and JDRF, and his scholarship has recently been recognized by awards from JDRF, American Institute for Chemical Engineering, and the Biomedical Engineering Society. Thanks so much for joining us, and we're really excited about this talk because it appears that you've kind of taken a new approach and meticulously gone through and appreciated or shared the appreciation of how these engineered cells may be phenotypically or transcriptionally different from stem cell, from in vivo beta cells.
Yeah, no, I appreciate the invitation to be here. And it's really been since 2020 when I was
Right.
last on the Sugar Science.
Yeah, you were in your basement and
Which is actually where I am today, by the way.
right.
Not much has changed in almost 4 years.
Yeah, and you were rushed to pick up your child at preschool, I think. But
Well, fortunately, it's the middle of the day, so no rushing at the end to go pick her up before I get a nasty phone call.
great.
Great.
Well,
Shall I go ahead and get started?
yes, please.
Great. Hello, everybody. Again, it's been a while since I've been on the Sugar Science, so I'm glad to be here again and to appreciate the invitation to share what we've been doing using single cell technologies to really understand and try to improve stem cell-derived islets for hopefully eventual use in therapy. My university requires me to make some disclosures about me having conflicts with regard to patents, employments, and the stocks and this stuff. So that's what my side here is doing. And so my talk, I broke up the talk into three segments, with the first one really just giving an outline of the use of SC islets, stem cell derived islets for therapy, for those who don't know about it, before really diving into the details of this publication that Mason Schmidt and I published earlier this year. And then I included some slides at the end on our ongoing efforts going beyond RNA sequencing to now multi-omic sequencing, if there is time at the still at the end of me talking about Mason's paper, but we can skip that if there isn't any time. So I'm not going to spend a lot of time on the background of diabetes because we're at the sugar science, so I assume that most people know about diabetes. But basically, the way that we view diabetes is from a very beta cell centric perspective. That diabetes is caused by there not being enough healthy, functional beta cells to keep up with the metabolic need of patients. And in the case of type 1 diabetes, that's because these cells are being destroyed by the immune system. Thankfully, of course, we have this wonderful miracle of a drug, insulin, which has saved millions of people over the century or so that we've had it. However, insulin is a treatment and not a cure, and also a very imperfect treatment. It's just impossible to administer insulin in such a way to uh, perfectly control one's blood sugar levels like a beta cell would normally do without you thinking about it. And because of these imperfections in the control of blood sugar and the administration of insulin, patients have a greater risk of death, lower life expectancy, and many other health issues and management difficulties that really is a very unmet need that we are trying to address with our use of cell therapy. And so basically the idea here is that instead of patients manually controlling their sugar levels through injections or pumps or sensors and all that sort of stuff, what if we just instead take islets containing the insulin-producing cells and source from an exogenous source and have these exogenous cells be transplanted into patients to replace the function that is lost in the endogenous cell. So this would be a cell replacement therapy or a cellular therapy and would be a functional cure for diabetes. This wouldn't address the underlying reason for why the patient became diabetic in the first place, but would at least provide a potential practical solution to blood sugar control. And so this is something that is done periodically through the use of islets that are sourced from deceased donors. So this is a figure that I adopted from a paper published by some colleagues of mine showing blood sugar levels before and after a patient with type 1 diabetes receiving islets from a deceased donor. And you can see really large improvements in the blood sugar control in this patient. So this is something that can be done and has been done maybe for a few thousand patients or so over the last couple of decades. But that's not a lot of patients, particularly when you look at how many people have diabetes worldwide. Again, not every single patient here, maybe it would make sense for them to receive a cell transplant. But there are a lot more people with diabetes than a few thousand. And as a bioengineer by training, I look at this and think that we really need to not rely on islets from deceased donors and instead need a large-scale source, a sustainable source of replacement cells and tissues in order to begin to meet the need for any fraction of people living with diabetes. And so the approach that my lab and other groups embrace on this front is using stem cells as this renewable cell source. So in particular, using pluripotent stem cells, which have the ability to replicate, self-renew themselves. This is where the scalability comes in, that we can grow up a virtually unlimited number of these cells if we give them the correct cues. Or if we give them different cues, we can instruct them to stop being a stem cell and differentiate into basically any cell type in the body, depending on which cues we give them. Of course, we focus on the cues and signals to instruct them to become insulin-producing beta cells and other cells that are found in the islet. And since these are cells that are grown in tissue culture labs and tissue culture rooms, these cells can be grown in bioreactors or other sorts of large surface area vessels in order to make large numbers of these cells, which do resemble islets. And so there's a lot of aspects that are under investigation in the field right now for how to make stem cell derived islets a actual real option for patients. And the biggest area that my lab focuses on is the generation of highly functional cells, basically trying to figure out how we can better give these stem cells instructions in order to become either more beta cells or better beta cells and um, better able to cure diabetes. And so, over the last almost decade now, we have published a slew of papers, first initially describing how to make stem cell derived islets, and then improving the process by which we are making these islets. And this presentation, we're not going to go through all these factors here today. These have been in the literature for a good amount of time. But basically, the way we go about doing this is really figuring out what are the correct chemicals to give these cells in what combination and at what timing in order to make the correct intermediate cell types that are shown here in a cartoon way, but also to best make the final stem cell derived beta cells that we can. And so I mentioned intermediate cell types. So we know the various markers that we're looking to generate here, mostly coming from the developed biology literature, including making sure that our process is very efficient at making cells that are expressing markers of endoderm, pancreatic progenitor cells, and endocrine cells, including C-peptide or insulin. So we've published protocols over the years and shown that we are able to make stem cell-derived islets from a large number of different cell lines, including human embryonic stem cell lines, like the Hughes-8 and H1 cell lines, iPS cells that we derive from non-diabetic donors, like 1013 and 1016, but also iPS lines that we've derived from patients with various forms of diabetes. Like this is a Modi patient. This is a type 1 patient. This is a type 2 patient. This is a patient with Wolfram syndrome, with or without the use of CRISPR to correct the mutation that gives them Wolfram syndrome. So you can imagine how having the ability to make these cells from islets from different backgrounds can lead to a lot of different areas of research, such as disease modeling, which I'm not able to talk about here today, but is a very active area of my lab as well. And so that's how the clusters look, kind of superficially like islets in culture. But when you actually measure the characteristics of these cells, they also do resemble islets in other ways that you would expect. For the final cell population, typically the majority of these cells are expressing insulin or C-peptide, co-expressing of them being the actual beta cell population in our system here. And almost all the cells are endocrine in one form of the other, including having somatostatin and glucagon expressing cells representing delta-like and alpha-like cells. These cells are able to secrete insulin in response to glucose stimulation with first phase and second phase kinetics here. So switching from low glucose to high glucose, you can see the insulin secretion response occurring within just a few minutes, like you would expect a normal islets to do. So we have this compared to islets that were sourced from a deceased donor. One thing you'll notice here, or become a theme later on in the presentation, is that while the stem cell derived islets are glucose responsive, able to secrete insulin in response to glucose stimulation, and also other secretagogues as well that I'm not showing here, they tend to do so in a manner that is reminiscent of being more immature, not as functional as human islets are, as represented in the differences in the curves here on this on this graph.
And Jeff, just to kind of like further that point, there is sort of like a window of immaturity you want them to be in as you place them in, a little bit less, regard to their oxygen needs and other things as they're implanted. Is that correct? There's sort of like a sweet spot for them to be in before they're being implanted into humans.
That is something that is theoretical right now. I know some people believe that with a pretty strong passion. I actually haven't seen any data to back that up.
Mm,
it may be true, but I'll tell you that even though these cells, from the oxygen perspective, since you mentioned that, even though these cells are immature versus human islets, they actually have a higher oxygen consumption rate than mature functional human islets. So the kind of some of the paradigms that exist in the islet literature from studying mouse islets or human islets, those
yeah,
don't necessarily always translate over to making sense in the world of stem cell derived islets. I'm not saying the idea of wanting them to be somewhat immature from a transplantation context is wrong. It's just that nobody that I've seen yet has demonstrated that to be a truth. And so, one of the guiding philosophies that I approach in my own research is that we don't know what the optimal characteristics and composition of these SC islets are in a transplantation context. And so what I'm trying to do is figuring out how to make the most mature, most functional islet. And once we have that as an option, we can go and start doing some of the more controlled studies of, how transplantation outcomes are affected by maturation, and we'll actually get a correct answer there. Even if the more mature cells are maybe not the ideal cells to be putting into patients, the new science and biology we would discover from actually bridging the gap in maturity right now would in itself, I think, justify the cost of achieving that.
great. That's great clarification. Thank you.
Yeah. And maybe to your point though, the even though these cells are immature versus donor islets, they are still able to reverse diabetes in a way that is comparable to human islets. And so here's basically taking data from two different publications and snapping it together, looking at stem cell-derived islets from a ES line, an IPS line, compared to human islets. And so in these cohorts, we had transplanted the same amount of cells to keep them all comparable here. And yeah, there's some differences, plus and minus a week or so in terms of the kinetics, but overall, the rate of cure and the set point that of blood glucose that these mice achieve are comparable to each other, getting this kind of human set point for fasting blood glucose levels, which is below the mouse's set points, which says that one thing that I like to see here. Um, uh, Again, with kinetics that are comparable to human islets that were also transplanted in this context. So we definitely don't need these cells to be perfect or maximally mature for them to have a effect. In fact, the clinical trials going on right now are, definitely not transplanting maximally functional cells. But I would suspect that if we're able to make these cells better, maybe more functional on a per cell basis, we could perhaps lower the dose that we need to achieve a cure, and that really helps out in terms of biomanufacturing. That each biomanufacturing run would make doses that can go to more patients if each individual cell is more potent, more functional. It also would make, I think, immune protection a lot easier. If you, at least conceptually, it's easier to protect 500 million cells from the immune system than it would to protect 1 billion cells. And obviously, I think if we were to take the extreme analogy here, if one islet, this is not, I don't think feasible, but if through the use of sorcery, one islet was sufficient to cure a patient with diabetes, this would be everything. We wouldn't be doing any more research in this area at this point, because the idea, like protecting one islet from immune rejection and manufacturing a lot of individual islets is something that's very achievable with current technologies here.
That would be very optimal.
So again, just getting back to the point about biomanufacturing. So since these are cells that are grown in the lab, they are amenable to conventional approaches for scaling up their manufacturing. And the system that we use in my lab now, which is really great for providing cells to collaborators, is that we're able to do the differentiation on high surface area flask. Getting a few billion cells in each one of these cell stacks, we can freeze the final stage 6 cells as a single cell suspension. And then as we need them, either internally or through collaborator requests, we're able to thaw these cells, aggregate them in either shaker plates or PBS bioreactors to formulate them back into living three-dimensional organoids, and then do experiments or send those cells to collaborators. And so this is something that is, quite nice and convenient compared to waiting for donor islets to become available, which is kind of hit or miss in terms of their availability.
Yeah, that's great. How many passages can those cells go through and still be looking, fabulous?
So we only passage them during the stem cell stage. They can go for a large number of passages. They sometimes will mess up. We have, like personally, I have gone for 50 or so passages and it can
Hmm.
work out okay. As a kind of a rule of thumb in the lab, though, we don't tend to do that and instead limit the number of passages of the stem cells from the thaw, just because, mutations can arise or karyotypical
Yep.
abnormalities could arise, and we just don't want to pay to need to constantly monitor that. And so we just address that by typically not going more than 3 or 4 passages before throwing the stem cells away and thawing a new bank of stem cells. Once they go through the differentiation process, there's no more passaging, and by the time they're at stage 6, even though we are single cell dispersing them, they are non-proliferative.
Mhm.
Because the endocrine cells and the beta cells don't really proliferate. And so we're freezing them and thawing and aggregating them, but there isn't really meaningful proliferation happening there anymore, which is uh, kind of a pain when it comes to practical use of these cells, but is, I think, a positive when it comes to the safety profile of these cells, so that since they're not inherently proliferative, unless we like really mess up the protocol and have proliferative cells that are a off-target population in there, they're generally going to be safe and not going to grow and form tumors upon transplantation.
All right. Well, that's great. Well, we'll have to talk a little bit more as we get into it about these off-target sort of population of SC islets with the serotonin producing cells.
Yeah, yeah, and you preempted that because my next slide, we're going to talk about that here in a second, because it does start to motivate the work that Mason had published, and some of the other work that I'm going to be talking about with ATAC sequencing if we have time.
Yeah, let's
So
just keep going as long as
yeah,
you've got. We'd love to hear it all.
Yep. Sounds good. So I've been basically focused here on the positives about how these cells can be generated. They resemble primary islets. They're functional, can reverse diabetes. However, there's a reason why we're still working on this, and that's because of the remaining challenges. And this really is the motivation for the paper Mason and I published in January, trying to reveal new insights about these challenges using single cell RNA sequencing. So the biggest two categories that stem cell-derived islets differ from primary islets on is in their function and their identity. I already talked a little bit about function. They tend to have weaker function, particularly in the in vitro assays. A thing that Monica just mentioned here is that there is also some undesired identity here, with probably the most well known being this population of serotonin producing cells that are either being inhibitory for the beta cell function, or at best case scenario are just there taking up real estate and lead to inefficiencies in the function of these cells by not actively positively contributing to the function and potency of the stem cell derived islets. So these are not cells that we want to have in there, regardless of what assumptions you have about them. But also, even then, the beta cells that we're making have immature gene expression, both in terms of having gene expression that is maybe higher than it should be for various on-target genes, but also little to no expression of common maturation markers of beta cells, such as MAPA and UCN3. And so one major area in my lab now for the last 4 or 5 years or so is asking the question, can we gain a better insight of these cells using single cell sequencing methods? And the reason why focusing on the single cell methods is that islets are inherently heterogeneous. And we had found it difficult to get useful information when relying on bulk assays because of this heterogeneity. So essentially, if you use like real-time PCR or just bulk RNA sequencing, you're essentially looking at a smoothie of the transcripts coming from the cells in the islet. Instead of using single cell approaches, this would be more akin to a fruit bowl, where you can actually discern each individual fruits that make up this smoothie. And perhaps we only, in this analogy, only care about the composition and characteristics of the strawberries of the fruit bowl. We don't want the other fruits in the fruit bowl to be affecting our study of the strawberries. And so a lot of groups, including us, have published over the years many single-cell RNA sequencing data sets of stem cell-derived islets from their own group, or data sets on primary adult beta cells or islets, or also one publication that included some human fetal beta cells. And one of the great things about the way that we do research nowadays is that these data sets are publicly available. Anybody can go and download this data. And so an idea that Mason, who's on the call, he was at the time a Center for Regenerative Medicine post-bac student in my lab, and recently moved on and is now a PhD student at Duke. And we also had a lot of help from Matt Ishihak in my group, who's a postdoc. The idea that we all had was, well, we have all this great data out there, and yeah, there was individual publications for each of these data sets there that were, valuable, and everybody should go and read them if you haven't already. But we were wondering if you can get any additional insights by taking all this publicly available data and putting it together into essentially one atlas of islets, that would span stem cell-derived islets, transplanted stem cell-derived islets, adult islets, and also fetal islets. And so when it comes to the data sets that are shown here for the stem cell-derived islets, these are 4 different data sets that come from 4 different labs that have 4 protocols that differ to various degrees, and also some cell line differences as well. So we have a pretty good mixture of different approaches and SC islet cell types here. So basically, the graph that I'm showing on the left is the UMAP of the cells that came out of each of these individual studies, and we're just showing chromogranin A to indicate where the endocrine cells are here. And you can see this with the kind of the reddish color here. Again, since it's a single cell RNA sequencing, what we're able to do is just focus on the endocrine cells. So we can pull out just the endocrine cells and there we go, and graph these together, no longer including or caring about the non-endocrine cells here. And if we just look at the endocrine cells, put all this data together from all these different publications, we're able to see populations that we would expect to see in islets. Now we have a population of beta cells that includes, again, stem cell-derived, adult and fetal, alpha cells, and some other smaller populations of endocrine cells as well. And these are just markers proving that, well, I'm not lying about what these cells are here. And so, again, now that we subset the endocrine cells, we could also subset and just look at the beta cells and start doing comparisons of, like, now we have all these beta cells from all these groups and all these different sources. How do they look? And they look incredibly different. There are several thousand, as you do all these pairwise comparisons, there are several thousand genes that are differentially expressed in SE beta versus adult, or SE beta versus fetal, or fetal versus adult. So even though these are all beta cells, if you look at it, from a high elevation view here, they all look very different from each other. And some of these differences help us to explain some of these questions that I had about the field. One of the ones I'm just showing here, because I think it's kind of cute, is that fetal beta cells are considered to be fairly functional, and we never really understood very well why they're non-functional. But like SC beta cells, have some degree of functionality, even though they're also immature, like the fetal beta cells are. And one of the things we found here in this meta-analysis is that we think that at least most of this is explained by the fact that some of the heterogeneity we see here in beta cell gene expression, some of that is the lack of instant secretion machinery in the fetal beta cells. For some reason, these stem cell derived beta cells, even though they're immature like a fetal beta cell is, they are at least getting the cues to know to turn on instant secretion machinery, which gives them the ability to have some degree of function. Not as good as adult, but this is why they have function and fetal beta cells don't have function. And so one of the other kind of more practical things that we did with this analysis is come up with a core beta cell identity that could be used to identify beta cells regardless of the context here. Again, being motivated by the large degree of heterogeneity in beta cells across different contexts here. And some of these markers are also shared to various degrees by other endocrine cells here. And so Mason did an analysis where he looked at each of these four contexts, listed out the genes that were enriched in beta cells versus all the other endocrine cells there, and then cross-referenced the lists across all four of these contexts. And he found that only 6 genes were consistently upregulated in beta cells across all these contexts versus all the other endocrine cells. And these markers here are not like surprising. These are very well known and understood markers. But the big takeaway here is that this is, I think, a very practical list that should allow for people to definitively know where your beta cell is under very diverse circumstances like what we're showing here. And it's something that my lab uses all the time now in order to be certain that we've identified beta cells in data sets, particularly those who are not living and breathing single cell RNA sequencing all the time and maybe have a little bit of a harder time identifying where beta cells are in newly generated data sets. So another question that we had here is we were wondering, how do these SC beta cells from these 4 different publications compare to each other and compare to adult beta cells? Because there's various claims out there about how more mature or less mature SC beta cells from a particular publication is versus prior literature comparison to adult beta cells. And what we found here is that despite these 4 data sets coming from different teams using different protocols, for the most part using different cell lines, and also spanning about 4 years of time or so, and when they were published at least, these cells are actually very similar to each other on UMAPs. They cluster with each other and away from the fetal and the adult. None of them are particularly correlating well with adult beta cells. And maybe the most important thing here is that these markers of maturation that are known to generally be low or missing in stem cell-derived beta cells, none of the protocols are actually achieving expression levels anywhere near or comparable to adult beta cells. So basically, this lack of maturity and lack of maturation marker expression is universal for all the protocols that we've looked at so far here. There isn't a particular approach that is at least represented by this data set that is meaningfully bridging the gap in maturation from a RNA level, at least. We did similar work with alpha cells and delta cells and see similar results with them. And also importantly, all the protocols are producing these serotonin producing what we call interchromatin-like cells. These do vary somewhat from each other, which you can see in the UMAP and the choice genes that I have here. But all protocols are having a problem with producing these cells, and we were not able to see these cells in adult islets or fetal islets. So indeed, there is something going on with in vitro differentiation that is causing these cells to appear, and that's nothing, none of this seems to be at least protocol specific in terms of the manifestation of these cells. So another major finding of this paper is that we saw evidence of there being persistent off-target and progenitor states in the actual stem cell-derived beta cells. Can we now subset down looking at just the beta cells, our strawberries and the fruit bowl analogy still? Here we're able to see that with all the data sets put together, able to see off-target and immaturity states being persistent here, such as having overexpression of MAFB, compared against adult beta cells, and also continued expression of PAX4, which is something that's typically turned off in adult beta cells. You can see this as well that in general for progenitor transcription factors that are basically expressed before the beta cell phenotype, SC beta cells tend to have consistent overexpression of these markers. So some of these markers we want to have there, some of we expect to have at a lower level in adult beta cells. Some cell-derived beta cells are consistently just overexpressing progenitor transcription factors, even though they are, beta cells here. Transplantation is able to help improve this. These overexpress genes are able to come down with transplantation, though there still is some difference in the transplanted stem cell-derived beta cells versus the adult beta cells by transcriptions, at least. And so this is true not only for individual markers like I'm showing here, but also in the inferred gene regulatory networks that would be downstream of these transcription factors. So this is a Regulon analysis, which is one way of inferring gene regulatory networks. And we're showing that not only are individual genes overexpressed, but also genes that are predicted to be downstream and regulated by these transcription factors are also the immature ones are also overexpressed. Again, such as PAX4 here being overexpressed in the SE beta cells. Within maturity, gene regulatory networks such as that of 6-3 is overexpressed in the adult beta cell. So it's not just individual genes that are problematic here, but it does seem that there is a major immaturity or misspecification of key gene regulatory networks that we're not able to currently overcome with in vitro differentiation. Another aspect of this was the large amount of neuroidentity that persisted in the stem cell-derived beta cells. This one was a pretty big surprise to me. I had suspected about the there being other immature gene expressing signatures, but the neuronal identity was something that really stood out to me. So one thing I like to joke about with my neuro friends is that neurons are just basically confused beta cells, and we're basically doing neurology as well. And the reason I say that joke that is has a mixed reception sometimes is that islets and neurons do share a lot of developmental transcription factors. That's why you have like neuro D1 being expressed in islets and islet 1 being expressed in some neurons. And so one analysis that Mason did here was to look at transcription factors that are shared in both islet and neurodevelopment, and saw that these genes tended to be expressed at a higher level in stem cell-derived beta cells versus adult beta cells. And again, this is true for individual examples like shown at the top here, but also true of regulons. That regulons that are enriched in neuronal targets tended to have higher activity in stem cell-derived beta cells versus in adult beta cells. So again, this seems to be some sort of issue with in vitro differentiation, that the cells are maybe going off the rails a little bit and are having a bit of a neural identity that we don't know how to control at this point. So the summary of this is that, and again, there's a lot of details in the paper that I'm skipping over for the sake of time, and it's a really good read, a really good resource. Just the amount of like tables and data sets that Mason produced and is available on the BMC Genomics website is, I think, something that at least my lab will be mining from years on. and to develop new hypotheses. But kind of speaking about some of the more specifics here is that SC islets across time and labs and protocols and cell lines are quite similar to each other and all have issues of immaturity, all have issues of lack of maturation markers, and have some other kind of new kind of aspects of immaturity, such as neuronal like identity here. So I don't know if we want to take questions at this point. I did have some
Yes.
on ATAC sequencing because that's kind of what I think is the next logical extension of this paper.
Anyone is welcome to drop a question into the chat and I'll bring it up, but we would love to hear the last piece of this talk. Oh, here's a question. If you had to make a guess, why do SCEC cells arise in current protocols?
I um I have a lot of guesses. I don't know which ones are right. For the sake of this talk, I'm going to just choose one arbitrarily and blame issues with the chromatin state.
Hmm.
But of course, it becomes a bit of a chicken and the egg issue, though, because is the chromatin state already wrong at the beginning, or are they getting different signals from the differentiation process that is causing inappropriate chromatin remodeling that is causing this to occur? I don't know. But I'm saying chromatin because that's what I'm talking about in the next section of the talk.
Okay, that's great. And then Nidesh from University of Alberta, how many max cells cryopreserved per vial and frozen as single cells before or after S6? And what condition, CRF or snap freeze?
Um, I'm not good with the exact numbers because I'm not usually in the lab nowadays. I think for a lot of the smaller scale differentiations, if we're not spanking a large number of cells, I think it tends to be around like 5 or 10 million per 1 milliliter of cryoprotectant media. When we're trying to do larger scale, I think we don't exceed 50 million. So anywhere between 5 and 50 million would be kind of routine for us.
Okay, great. And then Alexis Karmer, are there issues of immaturity and neuronal identity more concerned for function or safety?
I don't think I'm not worried about it from a safety perspective. We don't see any evidence that they can, for example, transdifferentiate into something else that would be problematic for us. I'm thinking about it more from a function perspective, and kind of the engineer in me wants to have a final cell population or a final cellular drug product that is well defined, changes minimally upon transplantation, and it has well characterized high function. And it doesn't rely on the cells needing to self correct themselves upon transplantation. That allows for us to have much better release criteria for these cells and with a lot greater confidence before we transplant them, hopefully in patients.
Yeah, almost like an optimization of the protocols or a tuning prior to implant. This is Xiaoming Wang, whoops, there's so many. Do the EC cell express in human fetal pancreas?
I don't think so. I haven't really seen good evidence for that. I know Micah Sander had a Micah Sander had a paper a year ago showing a little bit of evidence for it. So there's maybe a possibility it can exist at a very, very low frequency. I'm not 100% convinced about that, but I'm not, completely rejecting that hypothesis as well. To the degree that we see it with our differentiation, that definitely doesn't happen. So there is not a large, because we have about maybe 20% of our cells, 15-20% that are like this. We definitely don't see those sorts of proportions appear during in development. So maybe it exists at a very small level naturally, but not to the extent that we are having it happen in vitro.
Yeah, so when you say you guys see a identification of substantial off-target population of the SC islets that are consisting of the serotonin producing cells, what is that percent? Do you remember?
It's about 15-20% typically.
Yeah, it's interesting because like in the gut, those cells, EEC cells are like only 1%.
Right, right. It's actually kind of hard to make those cells with in vitro differentiation for gut tissues, as opposed to we make it it's kind of like weeds in our protocol. They're
Yeah.
there, not valuable for us, but they're like, flowers for other people's gardens.
Yeah. Send them over to the gut people. Okay. So for Larry Spears, can chromatin remodeling issues be inherited? Does age race affect this? Differences between donors?
Yeah, so we don't have enough donors to be able to make determinations based on um, age and grade, age or race or other sex or other differences there. I wouldn't be surprised if it is different, at least across sex, but I don't know that for certain just because we don't have enough statistical power in our data sets to be able to delineate that. I mean, chromatin can be inherited. There is methylation patterns that can be inherited maternally. Uh, the process, though, of establishing stem cell lines like this tends to remove a lot of those marks, though. There's actually some labs that specialize in trying to preserve a particular chromatin marks for the cells that they're studying. Pluripotent stem cells tend to erase those, but maybe also gather other marks that are not seen in development. That's a speculation on my part, though.
Okay, and then great presentation, Dr. Millman. Could you please elaborate more on those 6 genes that are common across all beta cells? And do you think we should focus in trying to upregulate those in differentiation protocols for better products?
I don't know if those. Well, one of those genes is insulin, so having more insulin is probably probably a useful trait for cell therapy. But I don't think there's anything particularly special about those genes in terms of like them being a target for improvement in the differentiation protocol. I've been more thinking about it as a just practical combination of markers that people can look at in their own single cell sequencing data sets to have reasonable confidence in having identified your beta cell population. And I'm saying that just thinking about like practically working with these data sets. Oftentimes you'll go and send out your samples, you wait a month, you get the sequencing data back, you get really excited, you open up Seurat, you put it all in there, you do some basic UMaps, you look at it, and you have no idea what you're looking at. And it can be oftentimes, depending on the experiment, really difficult to know where your beta cells are unless you like really live and breathe single cell sequencing data. But a lot of times people are not, full-time in this. You might be like a third year PhD student. You might do, 1 or 2 single cell sequencing experiments in your entire PhD. And therefore I view this gene set to be particularly valuable for those people who are on the front lines. They don't live and breathe single cell every day, and they can just use this to have a good degree of confidence that those are the beta cells.
Yeah, it's an excellent tool that your team has provided. Do you want to, here's another question, Ziming Wang, do you want to plan to get rid of EC cells in the future beta cell protocol?
Yeah, so that is one of the design goals that we have in the lab right now. And basically, most protocol, but most lines of research in my lab that have the potential to improve the protocol. One of the ways we define success is by improving the beta cells, but also lowering or hopefully eliminating the interchromatin like cells. Like I said, at best case scenario, they're just there taking over space that could be used for having more beta cells. At worst, they could be maybe dangerous themselves, or be giving inhibitory signals that will make the beta cells you do have there be worse. And so that is, I think, if protocols were able to get rid of them, that would be a major advance in the field.
Yeah, they're very weird cells. They're like sensors of the luminal nutrients. They're almost like a more primitive, kind of thing. They're in the mouth, the lingual area. Anyway, so that's it for another talk. But
Hmm.
here's another question. You have a lot. Thanks for the great presentation. A question from the publication. Could you comment more on the higher expression of ribosomal genes in SC beta cells? How do you balance the finding with the inherent issue of several single cell studies regarding ribosomal expression as a technical artifact? Ooh.
Yeah, I mean, one weakness and limitation of this study is that we were relying on on data sets that were out there. And so there are differences in technical details of how the data sets were done. Like we weren't there to look at people getting these data sets to ensure that there wasn't some sort of issue that could cause changes in ribosomal expression. So it's something is kind of unanswerable. We decided to include it in there and add some text around it because it was a finding, and it was something that really stood out to us. And so it could be a technical artifact. It's impossible for us to get to completely eliminate that possibility. If it does represent biology, one of the thoughts that we had here is that for the stem cell-derived islets in particular, one of the things that they are doing still is producing a lot of hormone that they are trying to store up. And so we were thinking that perhaps that might be an aspect of the immaturity here. The beta cells are trying to build themselves up, have a good store of insulin, and perhaps need to have more ribosome there in order to allow for them to build themselves up to being close to adult-like in terms of how much hormone they had stored.
And here's the last one before we just do a quick run through the last bullet point. Ding Yu says there are some old literature claiming beta cells could express serotonin under certain conditions. Do you think the off-targeted SEEC cells are more like intestinal EEC cells or like serotonin expressing beta cells?
No, that's a really good question.
Mm.
So I'm going to go in reverse order here. These are not a intestinal off target, as in we are like at least for our process in the lab in my group, we are not going through a intestinal progenitor cell. I could say with confidence that these interchromatin-like cells are coming from a pancreatic progenitor cell. So that's one thing that is important here. I'm sure that you can get intestinal off targets, and if there are intestinal off targets there, could they become serotonin producing cells mixed into your population? Yeah, that is a possibility. But at least for the way our process works and how efficient our process is towards making pancreatic endocrine, we are not making intestinal progenitor cells. These cells are not coming from an intestinal progenitor cell. With that said, yeah, there is literature out there about the ability of human beta cells and mouse beta cells to express serotonin at a very low level. I think there's one paper I've seen that they used some really fancy high sensitivity method to detect that normal adult beta cells do secrete a very small amount of serotonin. There's also another paper, I think this was done in mice, where they said that maternal beta cells in pregnant mice have an upregulation of their ability to produce serotonin. So there is literature out there that there is some serotonin production that can occur in beta cells. And probably what's happening here is that this kind of natural ability for the cells to produce a little bit of serotonin is being somehow exaggerated or enunciated through in vitro differentiation, that we're accidentally turning on this feature of the beta cells that are usually on at a very, very low level.
Thank you so much. Do you think you have time to just run this?
Yeah, I can skip and maybe just do a few key slides here.
I think people would really enjoy it, and there's quite a few people on the call.
So one thing we had a publication last year, and we're doing a lot of follow-up experiments with this right now, is creating data sets now using single-cell multi-omic sequencing. Since we saw such huge differences on RNA sequencing, we were really wanting to understand how these cells were also being affected through chromatin accessibility, with the idea being if we had this extra dimensionality of the data that could help us to, again, better understand in vitro differentiation and come up with better hypotheses for what's going on and how we can improve the differentiation process to overcome the challenges that we have right now in stem cell-derived islet differentiation. So, getting back to the smoothie analogy here, so, RNA sequencing maybe is the color, and ATAC sequencing would be like the taste of it. So we maybe care about like our strawberries and how sweet our strawberries taste and how red our strawberries are, good chromatin and good transcriptional identity of these cells. And so in this paper from last year, we had done multi-omic sequencing comparing, the stem cell-derived islets versus isolated adult islets. And we're able to, I'm going to skip some of this, again, identify the beta cell population, do a head-to-head comparison of the beta cell population. This is just looking at RNA right now. And this is data that is similar to what I already showed you with the that was in the BMC Genomics paper, that the primary beta cells, had higher expression of maturation markers such as UCN3 and MAFA. The SE beta cells had higher expression of other markers that are not associated with maturity. But now with this study, we're also able to pair transcriptional information with chromatin information. In particular, what we had found to be very useful in this initial study from my group is looking at the chromatin accessibility of transcription factor DNA binding motifs. So transcription factors have particular motifs that they have affinity towards on the DNA. But if the chromatin is closed, then the transcription factors can't physically get to the DNA to perform its transcriptional regulation capacity. And so you would infer if the chromatin is open at the motif, that the cells are ready to receive the transcription factor to do its function. But if the motif is closed, then it doesn't really matter too much if the transcription factor is there or not. The cells are not able to respond to those cues. And so here we see very large differences in the chromatin of SE beta cells versus primary beta cells. But again, primary beta cells having motifs associated with maturation, like June Phos, pretty important for that. But the stem cell-derived beta cells, surprisingly, having large open DNA binding motifs for progenitor and off-target identities, such as like intestinal identity, CDX2, LMX1A is expressed by the interchromaffin cells. SOX9 is a progenitor marker that is turned off in beta cells. SOX2 is a stomach marker in this context. So even though these beta cells actually don't have the transcript for any of these four genes that I pointed out here, they have open chromatin showing they're ready to receive that signal. It's not happening because the transcription factor isn't there. But from a chromatin perspective, these cells are primed to be immature and have the issues that we have here. So it's not just a matter of where they're at transcriptionally, genomically, or basically from an epigenetic perspective, these cells are also primed in a way to be immature and to have off-target identities. And one of the things that we proved in this paper is that by specifically trying to target chromatin regulation, we're able to, in this example, I'm going to show you, make the cells a bit better. And in another example, I'm going to show you making the cells a lot worse. And so one of the most differentially expressed genes here is this gene ARID1B. It's a well-known chromatin regulator. Here, we're able to show that knocking down ARID1B would result in some improvements in the identity of the beta cells based on RNA, insulin content, insulin processing, and also can help to correct some of the chromatin errors or immaturity markers that we had here, such as reducing CDX2, the intestinal transcription factor motif. And so this is, I have a lot of other slides that I don't think I have time to get through here, but basically I wanted to end talking about ATAC sequencing, because I think that's the future direction here, that RNA is only part of the question. And as this technology becomes more available, there's more data sets being published out there, I think that we'll be able to basically have a follow-up study that includes both RNA and ATAC data of SE islets and primary islets to have greater insights into the why of why these cells are mature. I'm going to skip through all of this. And
I think we're going to have you back for another talk.
Great. Well, yeah, so that's the feature that I think we have here. I see we have one more minute, so I just want to quickly acknowledge, the support that we get from our donors, and of course JDRF and NIH and other institutions. So many great collaborators that we're able to work with to go and answer questions here. But of course, most importantly, my team here at WashU, with a good mixture of bioengineers and biologists that allows for us to do all the work that we are doing here, including a lot of single cell sequencing work. So thank you to the team.
And thank you so much. This was such an interesting paper, a really great talk. And also very appreciative to you thank you for making this talk accessible really across disciplines, career stages, opening up sort of these ideas for trainees, clinicians, all to kind of start thinking about these new forays that you guys are at the, you're at the frontier of, really bringing all these tools to bear and helping to really finely define the best possible HSC islet for implantation in humans. So thank you very much. And we won't wait too long again before we reach out for another talk with you or your group.
Hopefully less than 4 years.
Yeah, no, oh, I don't think so. I don't think we're going to, because you're moving at a really fast clip here and we want to be kept apprised. So I'd ask or I'd suggest that, we ran out of time for questions, but if anyone has any more, to reach out to you directly or to your team on the paper and ask specifically.
Great. Well, I appreciate the opportunity.
Thank you so much. Appreciate it. And have a great rest of your day.
Yeah, you too. Thank you.
Bye-bye.